Now, cholesterol, biosynthesis of cholesterol. What is cholesterol? Cholesterol exclusively found in animals, hence it is also called as animal steroids. The total body content of cholesterol is about 70 kg, that is 2 kg per body weight. It is amphibitic in nature, means it have both hydrophilic and hydrophobic molecules in nature. It is essential for life to perform important functions. Functions. It is a structural component of all the cell membranes present in our body. Synthesis of all the steroids occurs with the presence of cholesterol. It is also essential for the synthesis of lipoproteins, that is the total protein component of the body. It is also essential for the synthesis of fatty acids, which are being needed for the oxidation to get the energy in liver. Now, synthesis. About 1 gram of cholesterol is synthesized per day in adult human beings. Almost all tissues participate in cholesterol biosynthesis. Largest contribution is made by liver, 50%, and 15% by the intestine. Enzymes involved in synthesis are found in cytosol and microsomal fractions of cells. Acetate of acetyl coenzyme A gives all the carbon atoms in cholesterol biosynthesis. This is the molecular structure of cholesterol, that is C27H46O. It contains 27 carbon particles, 46 hydrogen molecules, and 1 oxygen. Acetate molecule, that is CH3COO. The acetate is being contributed at carbon atom 1, 3, 5, 7, 9, 13, 15, 17, 18, 19, 21, 22, 24, 26, and 27. COO contributes to the remaining carbon atoms. For production of 1 molecule of cholesterol, we need 36 molecules of ATP. It means 36 molecules of ATP are consumed during this process. 18 molecules of acetyl coenzyme A are needed as a precursor, and 16 molecules of NADPH are required for energy purpose. Synthesis of cholesterol may be divided into 5 stages. 1. Synthesis of HMG coenzyme A, that is beta hydroxyl and beta methyl glutaryl coenzyme A, having 6 carbon atoms. Formation of malonate, production of isoprenoids, synthesis of squalene, and final is production of cholesterol. First, synthesis of HMG coenzyme A. Most of acetyl coenzyme A condense and form acetoacetyl coenzyme A. Then, 1 molecule of acetyl coenzyme A added form HMG-CoA, that is beta hydroxyl beta methyl glutaryl coenzyme A. This process is known as condensation, and this is similar to the formation of ketone bodies. Here, the point to be noted that ketone bodies are found in mitochondria, whereas cholesterol in the cytosol of a cell. Second stage is malonate formation. HMG coenzyme A reductase is the limiting enzyme. It is found in endoplasmic reticulum, and it reduces the HMG coenzyme A to malonate, and energy is supplied for this process by NADPH. Third stage is production of isoprenoids. Malonate is catalyzed in presence of enzyme kinase and converted to 3-phospho and 5-pyrophosphomalonate. Decarboxylation occurs and it forms IPP, isopentyl pyrophosphate, and another isomer of IPP, that is DBP, dimethyl pyrophosphate. Both the isoprenoids, they having 5 carbon atom units. Fourth stage is synthesis of squalene. Units of IPP and DBP containing 5 carbon atoms further condense and form 10 carbon atom group known as GPP, geranyl pyrophosphate. Now 1 mole of IPP combines with GPP becomes 15 carbon atom and 2 GPP condense to form squalene, that is 30 carbon atom molecule. Fifth stage is production of cholesterol. Cholesterol production occurs by the hydroxylation of the product squalene and utilizing the oxygen molecules and NADPH, and result is conversion of lanosterol. Conversion of lanosterol to cholesterol involve 19 enzymatic reaction. It is a highly energetic reactions. Few enzymes which are consumed in this process are lanosterol, gymosterol, desmosterol, etc. This is the flow chart of cholesterol synthesis. Acetyl coenzyme A, group 2 molecules, they form HMG coenzyme A, then malonate, then isoprenoids of 2 building blocks, then squalene, the 15 becomes, and they combine with each other, they form squalene, and the squalene is being formed, converted into the lensosterol, and lensosterol finally converts into the molecule of cholesterol. Key points to be remembered: carbon atom 30 to 27 occurs in the synthesis of cholesterol. Removal of 2 methyl groups from carbon atoms 4 and 14. Shifting of double bonds from carbon atom 8 to 5. Reduction in double bonds C24 and C25 is the final production of cholesterol molecules. Now, how it is being controlled? The limiting reductase HMG-CoA is found in endoplasmic reticulum, is the main controlling factor of synthesis of cholesterol. Few hormones, they influence the activity of HMG coenzyme A through cyclic AMP process, whereas insulin and thyroxine, they also increase the synthesis of HMG coenzyme A, and result is increase the number of cholesterol production. And product of cholesterol, its own synthesis also control the synthesis of cholesterol formation, that is feedback mechanism. Compactin and levostatin, fungal drugs, lower production of HMG coenzyme A. Now, total body cholesterol pool. Cholesterol, our friend or enemy? Cholesterol pool gives from 130 to 150 grams. Daily synthesis is 700 milligrams per day. LDL and VLDL, low density lipids, very low density lipids transport from liver to peripheral tissues as blood and known as bad cholesterols. They causes atherosclerosis, that is hardening of arteries. HDL, high density lipids, they transport from tissues to liver and they are good. They are also needed for the oxidation of foodstuffs. They are needed for the oxidation and to get the energy. The new research says cholesterols are our friends if we lead to a balanced lifestyle. The steroid ring structure cannot be broken down in human body. 50% of cholesterols are converted into bile salts, sauerkraut and collate, you know, and the extra cholesterols are being excreted in fecal matter. Cholesterols, they are serve as precursors for synthesis of all steroid hormones needed our body functioning. So I'm leaving this a question that I'm cholesterol. You can't break me once I synthesized. So it's up to you which kind of cholesterol you are going to synthesize. Thank you.